Hi class, we are moving on to the first two kingdoms that we mentioned in the first note. So we're going to do the Archaea and the bacteria. The Archaea is kind of a quick portion of this note because they are your prokaryotes, which means they don't have the genetic information in the nucleus. They also don't have your membrane-bound organelles like mitochondria and chloroplasts, and they live in extreme conditions. So they're kind of harder to study because of where they're found and located. They do have specific cell features that allow for them to live in these extreme conditions. So there's 5 types of archaeas. There's the thermophiles, acidophiles, and the halophiles. Those are the first 3 we're going to go over, and then there's 2 more on the next slide. So the thermophiles live in very high temperatures, so extremely hot temperatures, anything over 100 degrees Celsius. Acidophiles need to live in a pH lower than 3, so on the very extreme acidic side. Halophils, you're looking at your high salt concentration. So anything above 20% salt concentration, which is very high. The methanogens, your methogens, are going to need low oxygen, so they survive better in environments where there's less oxygen, so that what they're able to do is they're able to do their cellular respiration or their metabolism, and they actually create your methane gas. They're going to be seen sometimes in those digesters that break down animal and food waste so that they can make methane gas and burn it. And so these kind of archaea are actually found there. And then the last one is your psychrophils. They are going to live in the Arctic or the Antarctic, so they are living in those extremely cold temperatures. So for the Archaea kingdom, all you really need to know is that you have your prokaryotic cells that live in the extreme conditions and the 5 different types. Okay, now we're going to move on to your bacteria kingdom. Again, you're having your prokaryotic cell, just like the Archaea, except for these ones are going to have a little bit different cell structure because they don't need to survive in those harsh environments. Okay, so some characteristics of your bacteria. They have DNA, so you're double-stranded, but it's going to be in one circular chromosomes. Where we are going to have your 46 chromosomes, they are going to have one circular. Because it's a prokaryotic cell, it doesn't have a nucleus. It's going to have this little small loop of genetic information called the plasmid. And you'll if you ever looked into genetic engineering, you'll see that a lot kind of pop up. It's going to have a flagellum, which is kind of this tail-like structure here that whips around for movement. It has these pili, which are allowed for transfer of genetic information when the bacteria cells need to do that. So bacteria can also have different shapes. There's going to be 3 different shapes. You have the cocci, which is spherical, bacillus, which is your rod, and Spirilla, which is your spirals. And those have to do with the actual names of different bacteria. If you think about the strep virus, so that cause your strep throat, it can cause impetigo, things like that, it's called streptococci. And so what that means, and you can see it down in this picture here, is it has the spherical shape, and because that strep's in front of it, it means there's multiple of it. And so based on the actual name, you can kind of figure out the shape of the different bacteria. How do bacteria do metabolism? So how do they get energy? They can be autotrophic or heterotrophic. And if you remember back to grade 9 ecology, autotrophs make their own food, heterotrophs need to obtain the food. And so bacteria kind of can do metabolism with oxygen, without oxygen, or it can adapt to whatever it needs to based on their environment. So you have your aerobic, you have your anaerobic, and then your ones that can do both. When bacteria need to reproduce, they primarily reproduce asexually through a process of binary fission. And if you kind of look at the picture here, it looks a lot like mitosis, where you start off with your 1 parent cells, you duplicate your DNA, and then the cell splits into your 2 daughter cells. Those 2 daughter cells are going to be genetically identical. There's other ways that bacteria can reproduce depending on the conditions. So binary fission is usually the main one they do. They can do conjugation. That's when the conditions aren't going to be as favorable, so binary fission can occur. And what happens is that they are going to attach to another bacteria and transfer some of the genetic information. And so by doing so, if that bacteria can no longer survive, it didn't lose their genetic information. They actually passed it on to another bacteria cell. Transformation is the bacteria's ability to pick up pieces of DNA. Sometimes it's going to be, primarily it's going to be from a bacteria cell, but other times it could be from another species. And this is what's known as your horizontal gene transfer. And this is what they use when they're using genetic engineering and gene therapy. Endospores can also be reproduced, especially when the conditions are really severe and the bacteria cell is not going to be able to survive it. They actually become dormant in what's called an endospore. And what happens is, if you kind of follow through the picture, it's going to have your genetic information and then it gets surrounded in your capsid. And so this kind of very resistant structure goes around the genetic information and allows for it to stay dormant for a period of time until the conditions become ideal again and they're able to grow and function as a normal cell and keep going in its normal cell life. Why are bacteria play such an important role is there's lots of kind of good that come out of bacteria, not just your diseases that we kind of always know about. Bacteria play a central role in all of your cycles. So if you think back to grade 9 ecology, when you talked about your carbon and nitrogen cycle, the decomposers there are usually bacteria. And so they're able to break down or change different compounds to another compound so that they're more usable. They can be producers. So this is where they have the ability to do photosynthesis to put oxygen back into our environment. We have your intestinal bacteria, which helps break down foods as well as produce different vitamins. Food production uses bacteria, so how you make your cheeses and your yogurts, that's why you always have your probiotic yogurts. And then bacteria can also be used to make antibiotics. And we'll go through kind of a little bit about antibiotics in the last slide. The negative side of bacteria, we kind of always associate bacteria with your diseases. And so bacteria can cause different sicknesses depending on what type of bacteria is being affected. Strep kind of is one of the biggest ones. There's also tuberculosis. There's different bacteria that can just affect plants and cause plants to grow. There's ones that can affect food to spoil and go bad as well. So if you have, say, strep throat, you go to the doctor, they will be able to do kind of a quick test, tell that you have the strep bacteria, and from there they'll be able to treat it with antibiotics. So antibiotics have the ability to kill bacteria, but they will not treat viral infections with antibiotics because it won't do the same. So what happens is, say we have this petri dish of bacteria. You treat it with an antibiotic. All of the yellow ones here have been killed off. The one blue one is able to survive because it's resistant to that antibiotic. When it has offsprings, they are now going to be resistant to the antibiotics as well. And so doing so, you have now created this new kind of antibiotic resistant bacteria and the more you treat it with different antibiotics or stronger antibiotics, it's going to produce stronger and stronger offsprings. And so then these bacteria become harder and harder to kill. So sometimes you will see doctors being less resistant to give antibiotics out as much as maybe patients would like because they're trying to prevent this antibiotic resistance happening. And so you're trying to make sure that you're only using those antibiotics when you absolutely need to. Okay, that's kind of the end of the note. Just make sure that you go and do the worksheets for both of these. Okay, thanks class.